Hello everyone, today our group will be talking about the mechanism of muscle contraction. First of all, we will be describing the structure of neuromuscular junction. Secondly, we will be explaining the transmission at the neuromuscular junction. And thirdly, we will be describing the structure of the sesame. So first of all, what is a neuromuscular junction? It is a junction between a motor neuron and a muscle fiber. The neurotransmitter that will be involved is called acetylcholine. As we can see here, this is a motor neuron, the yellow one, and this will be the muscle fiber for the peach one. There are three components that we have to know for this structure. First of all, we have to know what is a synaptic terminal of a motor neuron. Synaptic terminal has a synaptic vesicles that will contain a neurotransmitter, which we know will be the acetylcholine. So secondly, we'll be talking about the synaptic cleft. What is that? It is a gap between the synaptic terminal of the motor neuron, which we can see here as the yellow one, and the motor end plate, known as the sarcolemma of muscle fibers, which you can see here with the peach color is the motor end plate. So this gap over here, we call that as a synaptic cleft. So now we will be talking more about the motor end plate. What is the motor end plate? As we read just now, it is a sarcolemma of the muscle fibers. What is a sarcolemma? From what we can see here, this is what we call a folded sarcolemma. It's like a hair root. It's like a root. So each extensively folded sarcolemma on the motor end plate is called junctional folds. So kalau ada banyak-banyak nih, it is called as a junctional folds, and they have a ligand-gated ion channels, so that the neurotransmitter here, they can also known as the acetylcholine, can go inside to the muscle fibers through the folded sarcolemma.
Now we will look into the transmission of impulse in neuromuscular junction. So here we have neuron, it has a terminal. And also here, is the presentative membrane. Between the neuron and the muscle, there is actually a very small gap between them. So the neuron will pass on the action potential to the muscle by releasing chemicals of neurotransmitter. So now let's zoom in here and see what happens. As you can see here, here is how it looks like using the part that I suppose just now. And outside here, you have the calcium ions. Okay. And also the plus ion. So the arrival of the action potential at the axon terminal, which is here, will depolarize the presynaptic membrane and opens all these voltage gated calcium ion. So now it triggers the calcium ion to move in, which causes the influx of calcium ion. So now inside here we have the calcium ion. This one, the color one, is actually the synaptic vesicle. So the influx of the calcium ion causes this to move up and fuse with the presynaptic membrane, which is here. On the fuse here, they will release the acetylcholine into the synaptic cleft, which is here. The acetylcholine will released by the vesicle will binds to this specific receptor. This triggers the opening of ligand the ion channels, and this will allow in the past ion to move inside here. So, this will lead to a depolarization of the muscle fiber and trigger an action potential in the muscle fiber. Action potential is propagated along the plasma membrane, which is, as you can see right here. The action potential triggers voltage-gated constant ion channels to open, Ca2 plus release from the sarcoplasmic reticulum into sarcoplasm.
On to the next part, we will be talking about the structure of the skeletal muscle. So there are two types. So the first one, we will be talking about the myo and the sarco. Myo is a muscle, so ya muscle kita, and then while sarco is flesh, yang daging-daging kita. And then the second component will be muscle fiber parts. There are 5 parts. So the first one will be myofibril, as we can see here, myofibril here, ini. It is an actin and myosin. Jadi actin and myosin ini kita dua nih kita panggil dia one panggil dia myofibril. And then secondly we have sarcomere. It is a repeating unit of a myosin and an actin. And then we have a sarcolemma, a plasma membrane, a sarcoplasm, a cytoplasm, and a sarcoplastic reticulum. It is an endoplasmic reticulum that stores the calcium ion inside the muscle fiber for the muscle contraction. Let's continue with the zoom-in image of the muscle fiber. So the zoom-in image, we have for the muscle fiber, we have the nucleus with the light one band. The light one band and the light I band is located in the part of the muscle fiber. And other than that, we got the dark A band. Dark A band is of course located in the darker side of the muscle fiber. And then we got the mitochondrion, we got the sarcolemma. The sarcolemma is basically the plasma membrane for the muscle fiber. And then we got myofibril. Okay, so let's take a zoom in the myofibril. So in the myofibril, it contains of thin filament which is made out of actin, and thick filament which is made out of myosin. Yeah, so the myosin and actin are organized into repeating units called the sarcomeres. So here's the sarcomeres. It is made up of the repeating of the organized myosin and actin here and here. Other than that, we got the Z-disc, we got the H zone, we got the I band, we got the A band, and we got the ZM line. And the last one, we got the sarcoplasmic reticulum. So the function of sarcoplasmic reticulum is to store calcium inside the muscle fiber for muscle contraction. So let's go down here and take a look at the component of sarcomere. So in the zoom-in image of sarcomere, we have 5 components. The first one is the zone that consists only of thick filament, a lighter region on both sides of the M line. So the thick filament is here. The zone that consists only the thick filament is the H zone. So this is H. The second one is the band that consists of both thick and thin filament overlap. So the thick and thin filaments overlap is in here, so which is the A band. The third one is the band that consists only of thin filaments. The thin filaments is in here, so the band that consists of the thin filament only is the I band. The fourth one is the line that holds the thin filament and marks the boundaries between sarcomeres. So it is the Z line. The last one is the line that holds the thick filament, which helps stabilize the thick filament in the middle. So it is actually in here, but in this image it didn't show. As you guys can see, it's in here. Okay, so that's it.